1/4 of the population are carriers of Golden Staph. Many of us live with it in perfect harmony. It resides within our nostrils, and you may ask yourself, why would a bacteria choose to live in a potentially dry environment? Well, the reason is that it gives the bacteria an opportunity to disseminate. Think of the number of times you've touched your nose this morning. Then you go around and you touch all sorts of things. How many people did you shake hands with this morning? The bacteria can disseminate very, very quickly. Most of us have healthy immune systems that allow us to keep the bacteria in check. But what happens when we are unable to fight off the bacteria? Such as when we or our loved ones are sick or hospitalised, we turn to antibiotics. For more than half a century, antibiotics have provided the magic bullet to treat Golden Staph. However, we are beginning to lose the war against the bacteria. The problem is that our hospitals are ideal breeding grounds for drug resistance. They can be dangerous places to be. And we now have bacteria that are resistant to all of our frontline antibiotics. This recent data from the United States highlights the problem. Each year there are 1.3 million episodes of Golden Staph infections in American hospitals. Patients who contract the infection are 7 times more likely to die, and they generally stay in hospital for an additional 6 days at a cost of $10,000 per patient. This amounts to $8 billion to the American healthcare system. Unfortunately, 20,000 to 40,000 people will die of Golden Staph infections each year in the US. This is clearly unacceptable and highlights the fact that our current arsenal of antibiotics are losing their efficacy in the clinic. Unfortunately, over the past 20 years, we've seen the numbers of drug-resistant bacteria increasing in hospitals all over the world, as is shown in this graph that was taken from a recent edition of the journal Nature. At the same time, the number of companies that have been working in early stage research and development into new antibiotics has been steadily declining. The result is that our pipeline of new antibiotics that are essential for dealing with this healthcare crisis is becoming concerningly bare. This slide highlights the fact that over the past 30 years, there have only been 3 new first-in-class antibiotics approved by the FDA. Many of the antibiotics that we currently use were identified in the 1940s and 50s, which was the golden age of antibacterial discovery. Many of the drugs, such as penicillin, have been in use in the clinic for decades, and their chemical structures have been subtly modified to extend their clinical lifeline. A prime example here is the penicillin derivative methicillin. However, the bacteria readily find ways to overcome these new agents, as has been seen with the development of methicillin-resistant Staph aureus. So one widely accepted strategy to combat drug resistance is to identify and use new antibiotics in the clinic. Classes such as Pfizer's Zyvox, Cubicin and Wyeth's Tigersil are highlighted here. So our goal is to address the global shortage of antibiotics. Our research is focused upon the discovery of novel classes of antibiotics with the ambition of developing these into products that will be effective in the clinic. The initial focus for our research is on Golden Staph, but we do have a platform technology that allows us to work on other pathogenic bacteria and fungi. In order to achieve this, we have assembled a strong multi-disciplinary team that consists of medicinal chemists, biochemists, structural biologists and clinical microbiologists, as well as collaborations with experts in drug formulation and the development of animal infection models. So clearly, one important strategy to combat drug-resistant Golden Staph is to use new classes of antibiotics. Now, what do we mean by a new class? What we need are new chemical entities that work through novel mechanisms that have not been previously explored by other antibiotics. These new classes are required to combat drug-resistant Golden Staph, as they are likely to be immune from the resistance mechanisms that the bacteria currently employ. An important point to make here is, like many other drugs, the antibiotics do have a limited clinical life. However, physicians are quick to embrace new products if they deliver improved outcomes to their patients. Therefore, new products can quickly capture a sizable share of the market. This is highlighted in this slide showing the sales data for the 3 first-in-class antibiotics that I discussed earlier. Over the past 5 years, the market for these products has trebled to over $2 billion last year. Pfizer's Zyvox, considered to be the best in class, achieved annual sales of over $1 billion only 4 years after its release. And Wyeth's Tigersil, the most recent new product, captured 12% of the market share in only 3 years. So what is our invention? Well, we have discovered a new class of antibiotic with efficacy against Golden Staph. This invention is a new chemical entity that works through a new mechanism of action that is completely unrelated to any other product on the market. Our compound, known as a biotin triazole, as this structure shows on the slide, provides a chemical scaffold upon which we can now derivatise new antibiotics. So our research is now focused upon the chemical optimisation of our lead structure. We have defined a target product profile for this agent and laid out a clear roadmap as to how we can achieve this. Our research is now working towards developing a candidate with efficacy in an animal model. Being a novel chemical class, we believe that we will obtain strong composition of matter protection on our intellectual property. And through this approach, it is our goal to establish a preclinical candidate for progression into clinical trials. Hopefully one day we will deliver a new antibiotic to the clinic that will help to treat Golden Staph and to make our hospitals a safer place to be. Thank you.